Hello and welcome to this video on why antibiotics work for some bacterial infections, but not for viruses. This is a point of some concern when it comes to antibiotic resistance in bacteria, unnecessary treatment in others, and yet another group has an argument about increased cost for medical care. Of these, only the first relates to this video. Let's start with a fairly common example. You develop a cold, runny nose, mild fever, and lethargy. You go to the doctor and they mean well, or in America need to justify their exorbitant fees. They prescribe you with antibiotics. In other cases, you acquire some other viral disease and, through misconceptions and misapprehensions, think antibiotics will help you. No matter how this happens, you are not going to benefit from antibiotics. It should be obvious from the name, given antibiotics work on bacteria. If you want to treat a virus, then certain and generally very specific antivirals must be used. To understand why antibiotics work the way they do, we must first go over a brief overview of the bacterium structure. Bacteria are somewhat similar to human cells. They have organelles, DNA, and the ability to reproduce. Where they differ is the cell wall or membrane. This is the first way we can easily differentiate the two main kinds of bacteria, gram-positive and gram-negative. The gram-positive bacteria appear to have a purple colour when stained with gram or crystal violet stain, while the gram-negative do not. Common gram-positive bacteria are your Staphylococcus aureus and Streptococcus. Common gram-negative bacteria include meningitis, gonorrhea, and E. coli. The reason they stain differently comes down to one thing: the cell wall. Bacteria have a cell membrane, just like a regular human cell. Then outside this, they have a cell wall. A major component of this cell wall is a thing called peptidoglycan. In gram-positive cells, it can be up to 90% of that cell wall. In gram-negative, it can be as little as 10%. When you compare gram-positive to bacteria, the gram-negative bacterium have a much more substantial external membrane. That's yet a further layer beyond the peptidoglycan membrane layer. This outer membrane surrounds the peptidoglycan and is what makes it gram-negative. The stain simply can't get into it. Further to the peptidoglycan, there are other things that play a role. In gram-positive cells, there are teichoic acids. These are molecules that run parallel to the peptidoglycan sheets, and they are only found in a smaller subset of gram-positive bacteria. When you change focus to gram-negative, because they don't have the same substantial amount of peptidoglycan, they have other molecules, notably lipopolysaccharides. You may also see this with the acronym of LPS. These are part of what makes gram-negative bacteria much more toxic, as these LPS molecules are a form of endotoxin. Your most pathogenic organisms will often have these, and it's something that contributes strongly to their ability to become a pathogen. You'll also find there are lipids found inside the membrane, just like most other cells would have. Knowing this about the cell wall of bacteria, we can begin to look at specific antibiotic and antibiotic function. This is primarily because the antibiotics we tend to use are able to specifically target cell membranes or functions of the bacterium. The most commonly targeted functions relate to protein synthesis and cell reproduction or structure. The first of these are quinolones, and they're a class of antimicrobials that was derived from quinines. Quinines are used most often for infections by certain parasites. The way they work is they target the topoisomerase and specific versions of it. These topoisomerases are enzymes that are used during DNA synthesis and reconstruction. That's an important step when you want to make either more bacteria or more of anything else that involves the DNA. The quinolone will target different parts of the process, This will vary considerably between gram-positive and gram-negative species of bacteria. Some will more specifically target one version, such as Terpo4, whereas others will target gyrase. No matter what you're looking at, that's an important distinction. They have very specific enzyme targets. Rifamycin drugs further build on this, and these inhibit DNA transcription. Transcription is essential if you want to go from DNA to RNA and then protein. Nearly all of these are achieved via specific subunits in the RNA enzyme. They're made up of two parts, the 30S and the 50S. These are defining the size. The larger molecules the 50S and the smaller the 30S. When you block these, you can no longer synthesize RNA from DNA. What is particularly noteworthy with rifamycin class of antibiotics is that the RNA transcription cannot have gotten very far along in the process. In fact, it's only a couple of nucleotides in sequence, and this can no longer achieve its desired effect of inhibiting RNA strand initialization. Then we have other classes of antibiotics, notably those that look at damaging the cell's strength and structure. They do this by targeting the peptidoglycan. By beginning to damage the peptidoglycan, you more or less start taking away the structural strength of its cell wall. Because there's no structural strength remaining, osmotic pressure, or the water bearing down on the cell causes it to begin breaking down. It's the same way, for example, something that travels far under the ocean with all the pressure of the ocean will slowly be crushed by that force. When you start to damage the peptidoglycan layer, whether that's by preventing its creation or maintenance, you destroy the cell's structural strength. This is achieved by two main families of enzymes, transglycolases and transpeptidases. These are what build up and maintain the peptidoglycan that is the cell membrane. Just as with the RNA synthesis blockages, blocking these enzymes will prevent the cell, and specifically the bacterium cell, from keeping its necessary shape. Like many human cells, once the cell begins to fall apart and the bacterium ceases to have its proper shape and function, it will kill itself in a form of lysis. The two common forms of antibiotics that do this are your beta-lactams and your glycopeptides. These inhibit the synthesis of the peptidoglycan, or parts that are needed for it, and by doing so, result in changes in cell shape, size, and more. This can lead to exacerbation of cellular stress responses, and the cell will, as said, kill itself. In other cases, the change in morphology, such as what occurs with some beta-lactams, can then lead to the change in shape, and this change in shape can then trigger further cell responses that identify that the cell is becoming elongated and trigger lysis. Finally, we go back to the enzymes and ribosomes in particular this time. Enzymes is a very broad term, and it can be used for protein synthesis, converting of DNA to RNA, and more. The ribosomes are a specific organelle, and these are important, specifically because they have a role to play in protein synthesis. When you have the two subunits interfered with, and more or less making it impossible for the messenger RNA to pass through and begin building the protein sequences needed, you can't get protein at all. This is one reason why they are among some of the most broad categories of antibiotics used. These are generally going to fall into one of two categories, 50S inhibitors and 30S inhibitors. Now we know the broad process by which antibiotics work and different classifications achieve their result, we can see why these will not work for a virus. Antibiotics are meant for targets that are free-floating in blood or at least exposed to extracellular fluid. viruses are different from this. While they can and generally will spend some time outside a cell, that is where antibiotics might theoretically affect them, viruses do spend most of their time inside a host cell. And this is a place where they are relatively, if not completely, immune to any antibiotic effect. It simply cannot find the virus to affect it. This is even if the virus is free-floating. There are a range of reasons why the antibiotics will still not work. But in the first instance, the antibiotics can't get to a virus. The first practical and real-world reason is simply that a virus does not have the membrane or wall that a bacterium does. A virus has an envelope made from very different components. Using antibiotics against this is like using a metal detector to find plastic. You will get almost no response at all. The necessary components to create these different cell walls, membranes, and in the case of a virus, envelope, are so drastically different that the means used to interfere with one do not work with the other. Unlike bacteria, which attack your body's cells from the outside, viruses actually move into, live in, and make copies of themselves in the body's cells. Viruses can't reproduce on their own like bacteria does. Instead, they attach themselves to the inside of a healthy cell's genome and reprogram these cells from there to make new viruses. That is, they are hijacking the cellular machinery of your own cells or whatever other host organism there is and uses that. If your antibiotics were to be effective against them, it would also be effective against your own body cells and thereby cause wide-sweeping damage in the process of trying to clear away a virus. Unlike viruses, bacteria use their own enzymes to replicate. Viruses hijack the machinery of their host to replicate. That is the simple difference there. Even if an antibiotic was to target a replication process for DNA or translation processes to create protein, it would not affect a virus. The virus has no enzymes or topoisomerases or other necessary cellular organelles to be affected by the antibiotic. The differences between a human cell and bacterium means that you do not want to target the same enzymes and similar. That leads to the virus's replication and translation processes being protected from antibiotics, even if they could work in theory. Going back to the viral envelope and bacterial cell wall, a virus has no wall to speak of. That envelope is very different. Notably, there is a complete lack of peptidoglycan. Most of the viral envelope is protein, not a viable target for antibiotics which either punch a hole in the peptidoglycan or prevent its production in the first place. This also means that the mentioned mechanisms to destroy and lyse bacteria do not work for a virus. The envelope does not lose its shape or function when exposed to an antibiotic, and even if it did, the virus itself doesn't necessarily have the needed processes to undergo lysis. As you can see, there are some very basic differences between a virus and a bacterium, which make it impossible for an antibiotic to work against a virus. It does not matter how many anecdotes you have about how much better you may have thought it makes you feel. There is no physiological, biochemical, or cellular process by which that is true. In fact, it's quite the opposite. It could harm your immune system by decimating your intestinal flora. This acts as a means to help keep your immune system trained and maintained. That means it's more ready and able to respond to the viral infection. This is not to say that there are not some exceptional circumstances where through modification, conjunctive therapies, and more, an antibiotic may find another use in another capacity, but it is almost entirely impossible for an antibiotic as it is currently available to be used for a virus. Thank you for watching this video. If you have found it interesting, consider liking, sharing, and subscribing. Please post any comments, questions, or suggestions that you have below.